So before we move on to talk about some of the other genes that are affected besides the SOD1, I just want to give you a sense of how vast this disorder really is and how complex it is, right? So if we start with mutant SOD1 in the cell here, look at all the different effects that it can have, right? One, like we mentioned, is mitochondrial stress, right? That's what we talked about with respect to free radical generation. Mitochondrial stress can lead to activation of caspases, which trigger cell death. This mitochondrial stress can cause proteasomal stress, which can reduce how well things get broken down in the proteasome. ER stress, all of these stress responses, right, have to do with the inability to scavenge for free radicals. And so if the mitochondria doesn't work as well, if the ER doesn't work as well, if the proteasome doesn't work as well, right, that's what we see. We think that SOD1 plays a role in transporting various things down these microtubule tracts, right? And so mutant SOD1 disrupts axonal transport. There's so much that just this one mutation does in the cell. It becomes very complicated. Here's another really busy and complicated figure that deals with ALS, right? And so let's move and we'll talk about sporadic ALS now. And the genes that are involved in sporadic ALS, known to fall into three categories. So sporadic ALS. Again, there's 20-some genes that are going to be involved, but we can generally categorize them into three different areas. One would be impaired protein degradation. And maybe we should title this sporadic ALS genes, right? Because these genes are involved in each of these three categories. Impaired protein degradation, that's typically manifested by the proteasome or some inhibition of the proteasome. Impaired cytoskeletal transport. Or deficient RNA processing. Okay, so let's take a look at 2 or 3 more examples of genes and their associated proteins that are involved and where they're involved. So let's talk about, remember I said that TDP43 was the most common intracellular inclusion, right? And so we see TDP43 here and TDP43 here, and both the RNA processing and the failure of the cause of protein aggregation. Okay, the failure to break down these proteins, and so they tend to aggregate. So we know that SOD1 aggregates, but TDP43 will as well. It's also related to this protein called FUS, FUS, I guess is how it would be pronounced. And you can see that that's on this list as well. Right, so both TDP43 and FUS, and they're similar in what they do. So our example now we talk about is TDP right, and what these proteins typically do is they're nuclear proteins. So you find them in the nucleus most commonly. But in ALS, they tend to aggregate in the cytosol. And form these inclusions that I mentioned before. What they do in the nucleus is that they bind RNA. However, mutations allow them to aggregate, right? So when there is a mutation in the TDP43 gene or the FUS gene, right, something about their structure allows them to aggregate, and it allows them to convert, like we saw before, wild type protein into the type of protein that can then aggregate, right? It sort of takes a good protein and makes it bad. The result of the mutation, right? So mutant proteins aggregate. I mentioned that before. They also don't perform the same job of processing the RNA transcript. Right, that's the mRNA. And so you know that there's a lot of mRNA processing that goes on, right? That there's a 5' cap and a polyadenylation tail, and there's some splicing that happens with introns and exons, right? That's all RNA processing, and these proteins are intimately involved in those processes, right? And so now if we have mutant proteins, the mRNA that's being produced isn't going to be as useful to the cells. Okay, so that's one thought or one potential example of how ALS can be triggered is involvement of TDP43 and FUS. So if TDP43 is the most commonly seen cellular inclusion, protein inclusion in the cell, C9 ORF 72 is the most commonly mutated gene. Okay, and so you can find this particular gene in a lot of different places as well. All right, here it is with respect to mitochondrial dysfunction. Oh, and TDP43 was there too, as was SOD1. It's here in aggregation, so it's going to be another one of these proteins that can aggregate together. DNA repair, along with TDP43 and FUS, okay? So there is some redundancy in where we see these genes show up, but mediate a lot of different functions. So this gene, when it's mutated, manifests itself with hexanucleotide repeating subunits. So what are called hexanucleotide repeat expansions. And so we've seen these repeat expansions already before in Huntington's disease, right, where we saw CAG, CAG, CAG. It wasn't hexanucleotide, right? It wasn't 6 of them, it was only 3. But in this case, we're going to see 3 nucleotides repeated over and over and over. In healthy cells, we're going to see this nucleotide sequence repeated maybe 30 times. In mutants, we'll see this 100 times or more, okay, this repeated sequence. What this protein does typically is it's involved in membrane trafficking. So again, movement of material up and down the axon and getting it to the membrane. How it causes problems, right, is in the mutated form, RNA transcripts accumulate in the nucleus. So imagine why this might be. And so here's under this title of nucleocytoplasmic transport defect, right? There's C9orf72. You could imagine, right, if this cell is or if this protein is typically going to move or help shuttle RNA transcripts and traffic them out of the nucleus and down the axon or something like that. If this protein is mutated, it can't perform that function. If it's translated, right, those repeats, those hexanucleotide repeats, get translated into a toxic dipeptide. So these 6 nucleotides get translated into 2 amino acids that are linked together, and that species, that dipeptide, is toxic to cells. And then the final way that this becomes a problem, right, this C9orf72, is that there's just decreased wild type activity. Right, so maybe we could put that somewhere. I'll have to go up top here, right, that there's so in addition, right, to not in addition to those transcripts of building up in the nucleus, in addition to translation into this toxic species, it can't do its job normally because it's mutated. Okay, and that can lead to problems in the cell as well. Lastly, I'll talk about excitotoxicity in ALS. Ooh, it's getting bad. Excitotoxis. I gotta start over. There we go. My goodness. Excitotoxicity occurs when overstimulation due to glutamate release causes too much calcium in the cell. Due to how do I say this? Excess glutamate, the neurotransmitter, leads to large increases in intracellular calcium, and so much calcium that it's toxic to the cell. Okay, and so in all forms of ALS, right, excitotoxicity is a common theme. And so what we see in people who have ALS is that they have reduced expression of the excitatory amino acid transporter 2. And if you remember from chapter 6, or I can't remember, I think chapter 6, what this transporter does is it clears glutamate from the synapse, right? And it brings it either into glial cells or into presynaptic cells. Generally, it takes it back up into glia, though. So it takes that glutamate back up into glia. All right, so here under normal conditions, right, there's a whole bunch of EAA2 expressed, EAAT2 expressed, excuse me, and that glutamate that's present at the synapse gets taken back up into the glial cell. However, in ALS, right, what you can see is reduction in expression of this particular transporter. that means that the glutamate stays present at the synapse, and this glutamate activates receptors that allow calcium to come in, right? We saw that, certain types of receptors, NMDA receptors potentially, and somewhat AMPA receptors can allow calcium to come in. So decreased expression of EAA2 means more glutamate stays in the synapse. And this allows calcium in, and this calcium is what drives excitotoxicity. Okay, so there are drugs that help this, but modestly, right? There's a drug called riluzole. Riluzole prevents the release of glutamate onto this motor neuron here. But unfortunately, this only adds 2 or 3 months of life to someone with ALS. Right? Most people who die of ALS die because of the degeneration of the motor neurons. And for example, if the motor neurons that innervate, like the diaphragm, don't work, the person can't breathe. Okay, and then that is often the way that people who have ALS tend to die. Okay, so that's not probably the happiest way to end, but we did make it to the end. I think that's the best news of the day. If you have questions on any of this information, we will have a review session, and we'll do that on Tuesday night. I don't know if I mentioned that in the first video, and I'll get you a time for that too, where we can do that, or Tuesday afternoon, something like that. I'll get you the time, and we can do it via Zoom or Skype or Blackboard or whatever you guys want. Okay, thanks so much. So far, so good. This semester. Let's finish strong, and I'll talk to you in the coming days about the exam and the review session. Have a good day.